Is the screen visible? Okay. Right. So once again, good evening and welcome you to this live practice session for the Applied Environmental Microbiology course. I'm Guru Priya, Prime Minister's Research Fellow in the Department of Civil Engineering, IIT Madras. So today we'll be discussing about the drinking water microbiology mostly. Okay, so do you have any specific topics to be discussed? If you have, you can just put it in the chat box, otherwise given no. From any of the previous weeks content, from week 1 to week 9. If you have any doubts or any topic, you can just put it in the chat box. Okay. Okay, so yes, I'll. Do you have any other? If you have any other doubts, you can just post in the chat box at any time. I'll try my best to answer them. Okay, so this is the first question for today. Okay, there is B in the chat box. So I guess you all know about suspended solids and dissolved solids in water, right? So basically what we do is for suspended solids, we go for some treatment method um, where we can physically separate them. But any idea of what do we do with the dissolved solids?
We go for coagulation and flocculation.
Sorry, sir. I asked for dissolved solids. Like I was discussing about dissolved solids, then not the question which is displayed here.
Okay.
Okay, okay. We'll discuss about all these processes. I'll just show the answer for this question and we'll just see one more question and then we'll go for the discussion for this topic. So yes, as Parul Chowdhury has answered in the chat box, it's B for suspended solids. We go for sedimentation. This is a second question. Only there is a change in one word, so maybe it looks similar to you, but this is second question. I have asked which of the following process involves the removal of colloidal solids. Any answers? Okay, D. Okay, two of you have answered D. So, for dissolved solids also, I guess you told coagulation and flocculation, right? So, okay, let me explain this. Um, The answer for this is coagulation and flocculation. But before that, in wastewater treatment or water treatment, what we have is we have dissolved solids, colloidal solids, and suspended solids. In case of suspended solids or settleable solids, they can be easily settled. So what we do is we go for sedimentation. We just give particular dimension of tank and leave the water for a particular retention time, and the whatever can settle will settle. That is based on the specific gravity, size, all this of the particles. We identify some particles as settleable and suspended, and these particles can settle. Whereas what happens in the case of colloidal solids is these colloidal solids are generally they have some charge around them and their size is a bit smaller. So all they cannot like effectively settle down as such. So what we have to do is we have to destabilize them. For example, if our colloidal solid is negatively charged, has a negatively charged surface, there will be a mutual repulsion between all these particles because all of them are going to be negatively charged. There will be a mutual repulsion, and they will stay suspended in the liquid. So what we have to do to remove them from the aqueous system or the liquid system is we just have to break down this repulsion, or we have to alter the charge of these particles. So for that, in coagulation and flocculation, for what we will go is we will add alum, like we'll add alum or ferrous based coagulants. So what they will do is when we add a positively charged salt, the negative charge, generally colloidal particles are accepted to be negative charge, so the negative charge over their surface will be destabilized first of all. So this process is known as coagulation. And after that, because of overcoming of this mutual repulsion between the particles, now they can agglomerate together. So this agglomeration is known as flocculation. Once they agglomerate, there is a specific gravity or the density is going to increase, and they eventually get this capability of settling down or sedimenting. So after that, after coagulation or flocculation, we just allow them to settle, and then we remove these solids from the water system. But mostly in case of dissolved solids, what happens is dissolved solids, as we all know, they are going to be in utilizable form by a microorganism. Uh, Compared to a colloidal or a suspended solid, dissolved solid is easily uptaken by the microorganism because of there is no diffusional limitations and all. Whereas in case of this colloidal or sedimental particles, what happens is even though they are biodegradable particles, the organism has to produce extracellular enzymes in order to break this complex molecules into simpler form so that it can diffuse it and then consume it. But in case of dissolved particles, they are already very easily or more readily available for the microorganisms to take up. So they easily take up these particles, and then they convert. I mean, these particles are used for the growth of the biomass. So what we essentially do is the dissolved solids are consumed by the microbes, and it becomes a part of the cell mass for the cell growth. So we are converting it into a settleable particle. And after our this microbial process, what we generally do, we generally allow it for secondary settling, because I think we use this for secondary treatment processes. So it goes for a secondary settling tank. There this biomass settles down and we get a clear liquid. So this is how we remove this dissolved solids. Is the process clear, or should I repeat? I have discussed about almost all of the options given here. Is the process clear, or should I repeat? Okay, others, is it clear or should I repeat? I'll just wait for some two more responses. Maybe you can give a thumbs up if it's clear. Okay, okay, right. Okay, so that's the difference between suspended or settleable solids, dissolved solids, and colloidal solids. Each of the processes are used for each of these things. And finally, um, disinfection, we go to kill the microorganisms in it. In biological treatment, what we do is we use the microorganisms for biological conversion of the organic matter or to biologically treat the water. But in case of disinfection, again what we do is whatever some remaining microorganisms are present in it, we either go for chlorination or ozonation or UV based disinfection, and then we take this up. One more thing in this options is filtration can also like remove solids from water. Generally it is said to remove turbidity from the water, but in our option it is given along with chlorination, so this combination is not going to work out here. So that's why we are ruling out that. In case you have a doubt why filtration is there and why we are not considering it, filtration generally removes turbidity, yeah, fine, but here it's given along with chlorination, so it will not fit into either the first questions or the second question. Okay, right, I hope it was clear, and if you have any doubts you can post in the chat box at any time. I'll go to the next question. Okay, we have 2A and 2C. The answer is C here. It is rapid sand filtration. Um, Filtration, we all know it's a technique where we use some medium uh, to trap or stop the movement of certain molecules and allow the movement of others, generally in any random context. Okay, but in water what happens is we use filtration to remove as we discussed the suspended particles or turbidity in that case, and uh, after filtering out we will get a clearer liquid. We have sand filtration, we have membrane filtration, we have all these different kind of filtration. In sand filtration we have a rapid sand filtration as well as a slow sand filtration. In membrane filtration, as most of you would have known, it's microfiltration, ultrafiltration, nanofiltration, etc. So all these types of filtration are available. So in reverse osmosis is also a membrane process. So we use a membrane, and we know what happens is the movement of molecules based on the concentration gradient, but here it will be the reverse movement, so that's reverse osmosis. um, I guess you are clear that it's not biological removal, because obviously it's not a mechanical filtering process. But in case of adsorption is not a mechanical phenomenon, it's a surface phenomenon wherein physical and chemical forces are involved between the interaction. Like we have an adsorbent, which is the medium onto which something is going to get attached, and the adsorbate, which is the molecule that is getting attached. So the interaction between this adsorbent and adsorbate is usually going to have some um, physical forces like van der Waals force, or chemical forces like covalent bonding, etc. Okay, so it's not a mechanical activated carbon adsorption is not a mechanical filtering process. But when we carry out filtration, we can have activated carbon as one of the media for filtration. Like we have sand, we can also use this carbon. But there is a distinct variation between using activated carbon for adsorption as well as filtration. In filtration, the porosity or pore size plays a major role. In activated carbon adsorption, it's not the entire carbon population that is considered. What happens is we have this interaction between individual activated carbon entity and the pollutant entity. But in filtration, on the whole, the movement of the pollutant through the filtering system is blocked. Okay, so I think this is what is the point of confusion here. The main thing to remember here is adsorption uses physical as well as chemical forces. It's not like a mere mechanical filtering process. So here the example of a mechanical filtering process is a rapid sand filtration process. Is that clear or any discrepancies? Okay, okay. If you find any discrepancies with what I explain and what you have in your mind, you can just unmute your mic and let me know. The next question. Okay. Um, so, what is antibiotic resistance? Can you just give it in two, three words in the chat box? Maybe a single sentence definition or something. Resistance to disease. Okay. Okay, so all of you are right in one or the other way. So generally, as you have told, antibiotics is something that is used to kill microorganisms. Generally, it is like some chemical produced by one organism which kills the other microorganism. We know, for example, we know penicillin, ampicillin, streptomycin, etc. So all of these are few examples of antibiotics. When in the case of antibiotic resistance is the general role of antibiotics is to kill a microorganism, but when we give this antibiotic to a microorganism, when we expose the microorganism to antibiotic and it is not killed but instead it just grows there normally, that is when we term that the microorganism has gained resistance against this antibiotic, and this phenomenon is known as antibiotic resistance. It is of emerging concern uh, worldwide now, and it's one of the crucial research topic in the field of environmental engineering currently. Like many people are finding many ways to combat this antibiotic resistance, and there is also much of a civilian responsibility in combating antibiotic resistance. As we all know, we should not just discard antibiotics right away in some random land or water resources. We should not buy unnecessary antibiotics or buy them without prescription, buy overdoses of antibiotics. So all these are general public point of view. But scientifically, what happens is there is some gene that is responsible that confers this resistance to the microorganism. uh, there is also very much possibility that this gene get transferred from microbe to microbe, and considering a microbial ecosystem either in water or land, when there is this antibiotic resistance gene present in one microorganism, there are high chances of this gene getting transferred to a nearby microorganism, rendering that also resistant to this particular antibiotic. So this is a very much alarming phenomenon that is existing in the environment now. In the purview of this question, we have received mixed answers. Let's see the answer now. So option C is the answer. Let's see what all the statements one by one. So first one is antibiotic resistance is only a problem in hospitals and healthcare facilities. So as I told you, it's a civilian responsibility now, that it's no more a problem in hospitals and healthcare facilities alone. It's around us, it's everywhere now. be it land, be it water, or be it air. So we all know there are many microorganisms suspended in the air. Imagine these microorganisms getting resistant to antibiotics that we take. I think many of us would have personally experienced, maybe few years back, when we have a fever or some infection, the dosage of the medicine was different. It was lesser. Any antimicrobial medicine or antibiotic given to us was given in a very lesser dose, and we were able to recover from the disease with that dose. But now the dose has drastically increased. uh, Take any antibiotic or antimicrobial drug for that matter, we need to take a very higher dose nowadays to get the same effect or to get the same healing. So all of us have this experience of this problem. So it's not only a problem in the purview of hospitals or healthcare. As I told, when we are going to buy a bunch of antibiotics and when it's not used, we just throw it away like this anywhere. This can confer, like, I think we have studied about this selection process or some other process also, where we gradually tend to expose the micro for our favor. We gradually tend to expose the microorganism to some environment, and finally they are able to survive in some environment in which they are not able to survive before, right? So similar case here also, what happens is when the antibiotic is present in the environment randomly and the microbe is exposed to the antibiotic, it gradually gains the resistance or the capability to grow in the presence of that antibiotic also. So this is not only a problem in hospital and healthcare facilities, but all of us, uh, in all places it's present. The second option is antibiotic resistance is only natural with no human input and cannot be prevented. So, as I told you, we have a very big hand in this. We should not let antibiotic very randomly be present in the environment. That's because of the human intervention, this thing is happening, right? So, only because of this, there is a widespread of this antibiotic resistance or antimicrobial resistance present. Uh, and then the third option is overuse and misuse of antibiotics contribute to the development of resistance. This is also a part of the story that I told you. Obviously, overuse and misuse of antibiotics is a reason for developing resistance among a wide variety of microorganisms. Otherwise, in the lab scale, if we have to do it, we'll just do it in a very safe environment in biosafety labs basically, and discard it properly. So this doesn't go out of the lab environment, it's just for some research or scientific purpose. But because of this widespread antibiotic in other places all around the environment, antibiotic resistance is also very common everywhere. Okay, antibiotic resistance can be reversed by simply increasing the dose of antibiotic. No, because resistance is conferred by some gene. So gene, when it is involved, there will be some interaction, chemical interaction or a biochemical interaction basically. Because if it has to survive in the presence of antibiotic, the organism has to have some metabolite which is either expressed outside the cell or which helps in the metabolism of that antibiotic so that it can survive in the presence of that antibiotic. It's not like that. We just it's not only numbers. We just increase the dose of the antibiotic and the resistance is gone. No, resistance will not be reversed if we increase the dose of the antibiotic. Maybe the drug can work at a higher dosage, but the resistance will not be reversed at any case. Only some mutations or some other things can reverse the resistance, but not the increasing the dose of antibiotic is going to reverse the resistance, antibiotic resistance in microorganisms. Is that fine? You can either put in the chat box or give a thumbs up. Any discrepancies also, you can just unmute your mic and you can. Okay, got a couple of responses, so I'll just. Okay. So direct pathogen culturing may be a rapid process or a slow process depending on the nature of the microorganism. Because like when we are going to culture microorganisms like mycobacterium, um, mycobacterium tuberculosis is a bacterium that causes tuberculosis in humans. Okay, so mycobacterium is generally a very slow growing microorganism. So culturing that takes very long time. So in the purview of that, whichever is able to rapidly grow, we can culture that rapidly. So that will not fall as a limitation. It can detect both viable and non-viable microorganisms. I guess culturing, as you all know, will only show us the viable microorganisms, because essentially in culturing what we do is we just take our sample and plate it. Generally we use this petri plates, add a suitable medium like a nutrient medium like nutrient agar or endo agar or emb agar, depending on what kind of microorganisms we need to isolate. We add these nutrient media to the petri plates, and then we add inoculate the plates or the flasks, culture flasks, with this microorganism sample, and then see how it grows. So generally viable cells only can survive in culturing, non-viable will not. um, Being peculiar is not a limitation, obviously. So the limitation of direct pathogen culturing is it can underestimate the actual number of microorganisms present in the sample, because of the nature or the biochemistry that works in this process. As I told you, we cannot estimate the non-viable cells in the sample. So that's again going to be some underestimation here. So obviously it underestimates the actual microbial count that's present in the sample. I guess all the answers were right. So I'll move to the next question. Okay, as we have discussed, direct pathogen culturing was one method for detection of pathogens or microorganisms in any sample. We also have molecular techniques for detection of pathogens. Can you name some molecular techniques that are used for direct detection of pathogens in environmental samples? PCR, okay. Right, PCR is one technique. Uh, I guess all of you are familiar with PCR. It has been discussed in previous sessions. Okay, if I have to brief about PCR, can you just give me a thumbs up so that I'll know? So the participants from biotechnology, microbiology, or any such background would have heard about PCR. There are, if there is anyone who wants to know about PCR, just give me a thumbs up. I'll just wait for a few seconds. Okay, assuming all of you know what a polymerase chain reaction is, that is one of the molecular techniques. So the answer for this question is D. Let's, as usual, go with all the options. Capability to identify live pathogens. That's not a very big advantage because, as I told, in case of culturing, we get viable cells because only viable cells can grow on the plate or any medium that we provide. So that's very peculiar about culturing, that it can only give us information about viable cells. It can be advantageous in some case, disadvantages in some case. But in case of molecular techniques, we go for something like a PCR, where uh, in colony PCR, yeah, the cell grows. But in other kinds of PCR, as we all know, it's completely dependent on the DNA strands. So it's just PCR is basically the amplification of DNA, wherein we have this all the essential things for a DNA replication, like the primer or the nucleotides and other conducive environment for replication of the DNA to happen. So essentially, what happens is the DNA amplification occurs in a PCR, but uh, this doesn't like tell that it's only happening for a live pathogen, because DNA can be from anything, right? As we have studied about this gene transfer phenomenon, the bacteria can uptake a free DNA that's available in the environmental sample, which means the environmental sample in itself contains some uh, free DNA, even that can amplify and give us some results. So it's not like capability to identify live pathogen is not an advantage of molecular techniques. Similarly, molecular techniques are going to be costlier. Why costlier? In the sense, you maybe if you are working with a PCR or something, you will know what's the cost of a PCR mix, what's the cost of a PCR equipment, how carefully we have to handle it, all these things. Molecular techniques generally involve all these things that we can't see with our eyes. So obviously we have some calculations, we have some assumptions, and we have some estimations based on which we carry out our experiments. And then even in PCR we go for this gel detection after that in order to see our results. Depending on your purpose, after PCR, what you do with the PCR product is going to change. On a general context, after PCR, what we do is we run the gel to see what's there in it. So all this is going to consume a lot of cost, right from the PCR mix to how we are detecting the PCR product, or how you are quantifying the PCR product, either qualitative or quantitative analysis, both. All of this involves a lot of cost in it. So obviously, even other molecular techniques are not cheaper, they are costlier, so it's not low cost. And no specific equipment is required, not at all true, because even in PCR we know we need a dedicated equipment where we can run this PCR. So the only advantage is the fast detection of pathogens, because in case of culturing, as I told you, depending upon the generation time of the bacteria, you can have your culturing period from hours to days to even weeks. Like mycobacterium, as I told you, is a very slow growing microorganism, so that will take many days to grow, or even few weeks to grow. So that's a very painfully time taking process. Okay, so here in molecular techniques, that's not the case. Comparatively, it ensures us fast detection of pathogens. So that is the advantage of molecular technique. Is that fine? Okay. So I'll display the next question. Okay, so as you have answered rightly, both EMA and PMA treatments can be used. I'll just brief about this. So ethidium bromide monoxide and propidium monoxide, like these chemical treatment, when combined with PCR, they helps us to quantify the living cells in our sample. Both these are nucleic acid or nucleotide intercalating dyes. I hope you all know what is intercalation, which is generally binding to the DNA or RNA strands. So I think EMA and PMA both are like analogous to each other. They are both azide bearing dyes, and they can intercalate to our DNA or RNA. So when they are going to intercalate, uh, they are going to stop the replication process. So what happens is this DNA RNA intercalating dyes only cross the damaged lipid membrane barriers, which means only the dead samples they cross. So after that, what happens is they intercalate with the nucleotide, and then they, when we are doing the PCR, wherever the intercalation has happened, that gene cannot be amplified or replicated. Only the live things can be replicated. So this is one method to assess viability of bacterial cells in environmental samples. Here is the next question. What do they mean by biological instability here? You can also give that. and chat box. As usual, some one sentence with few words, 2 3 words. What is the problem of biological instability? Okay, so we have gotten a few responses for this based on the options, and also two people have answered for what is biological instability. It is a change in biological sample, okay, and it lose their integrity. Okay, the microbes lose their integrity. Okay, telling about biological instability, maybe people who are from um, microbiological or biotechnology background would have heard instability in terms of losing microorganisms. Generally we go for pure cultures, transport of pure cultures from one place to another. For example, when we order it for our laboratory purposes, we get it from somewhere where we get it uh, in packed form. We go for something called storage of microorganisms, where we go for dehydrating techniques like freezing or lyophilization, etc. So we do all of this because eventually we can't store microorganisms in normal environment. They will, as one person has told, they will start losing their integrity. But in the purview of some environmental sample, we know environmental samples are very rich in microorganisms. In this case, biological instability is a rampant growth or a very rapid or uncontrolled growth of microorganism in the sample. So even this is dangerous. Why this is dangerous? Because we all know this environmental sample is going, for example, a wastewater sample, let's take, is going to have some organic matter. So that is going to be a very conducive environment for the microorganisms to grow. And when it starts to grow very rapidly in that, at one point the nutrients are going to be exhausted. It's going to go to the stationary phase and the endogenous respiration phase, eventually the decline phase. Okay, so that is also not something that we will not prefer while transporting. So transporting of samples can be in different senses. I just wanted to give a overview of this instability can be of different senses, where you only transfer a pure culture, so you are worried about either doing a lyophilization or something, or when you're going to transfer a bulk like a environmental sample, in that this instability phenomenon affects in a very different way. Okay, I hope you understood different phenomenon in which this biological instability can happen in the microbial samples. So keeping that in mind, this losing of integrity mainly occurs because of the changes in the environment, as I told you. when we are uh, when we are going to change the environmental conditions like the temperature, the pressure, the pH, the other nutrition availability, or the accumulation of toxins, all this is going to affect the microorganism that is getting transported. So this will lead to mainly the uh, change is going to be in the protein. So like in a cell wall, cell membrane, or other proteins that are present, very essential for the metabolism or the survival of the organism, is going to get affected because of the change in environment, is going to cause a very significant instability in our sample. So this will be of not of our interest, because we need a safe transport to carry out the work with what we have to do with the sample. Okay, so keeping that in mind, all of these techniques are used in combating this biological instability. So as I told you, lyophilization or freezing, all these are low temperature techniques. We just go for dehydrating and maintaining it in a very low temperature, so that all the biological activity is arrested. We all know at very low temperatures this metabolism, I think in first few weeks of classes we saw about microbial classification based on temperature, where we saw about psychrophiles, psychotrophs, mesophiles, thermophiles, and hypothermophiles. If you would have watched the graph properly, maybe you can go back and watch it anytime, you would have seen that uh, just a minute, I think you, I'll draw the graph once. So this was our graph, where we had this temperature and growth rate on the y-axis. So we all know, roughly imagine this is psychrophilic, and this is going to be your mesophiles, and this is going to be your thermophiles. This part of the graph you all know. The x-axis, I think all of you are clear. You all know that psychrophiles will have their optimum around some 5, let's say. Mesophiles obviously here, and the thermophiles are they'll have higher temperature. But when you have a very close look at the y-axis of this graph, you can find that the psychrophiles will have their growth rate or mu lesser than this mesophiles, and the thermophiles are the ones that are going to have a higher growth rate on comparing with all of this. So what this suggests to us, if you cannot imagine this, or this is not clear with this graph, you can just go back to any microbial classification graph and you can view this. So this mu optimum, or the highest mu, is going to be lesser for a psychrophile than the normal mesophiles, and compared to a thermophile. We all know this bands of rule where it says for every 10-degree rise, the activity is going to double, something like that. While learning about enzymes, also you could have studied up to certain limit. When you're going to increase the temperature, the activity is going to increase. So that is what this suggests. Okay, so obviously, when we are going to reduce this temperature, the mu is going to reduce, so that there is not much of instability in the sample that we are considering it. Okay, so that is why we are going for lower temperature. When we take it to a higher temperature, obviously when you are going to rise the temperature itself, the activity is going to increase. And at one point what happens is, after this, maybe until this, for a hypothermophile, and until this point maybe we can see a increase in mu. But after this, eventually what happens is, because of a very high temperature that the microorganism cannot tolerate, all its proteins including the enzymes are going to get denatured. Maybe the cell wall itself may be denatured or damaged, and eventually its growth is going to come down. It's no more optimum, it's going to come down because of all these reasons. But in case of psychrophiles, your activity is a bit arrested, growth is arrested, and you can have a control over this instability phenomenon. Similarly, we can use chemical preservatives if we are going to be affected by something like a osmotic pressure thing, or maybe some chemical preservative which can arrest the growth of microorganism and keep it in a very stable metabolic state, that can be used. And also we can have it in a vacuum, because when we have air, this metabolism is going to be enhanced. But when we are going to have it in a vacuum state, the metabolic activity will be arrested in that state. So all of these techniques can be used for avoiding this biological instability during transport or during storage even. In case of storage, we mostly go for this low temperature storing. We store it in a fridge, or we store it in a -80, -20, depending upon the needs. We do all this cold storage things. Okay, with that, I think it was clear. Okay, so this is the next question. Okay, so as you have answered it right, I'll just go for a discussion only. Statement A is ingestion of contaminated food or water is the main mode of transmission of Giardia lamblia to humans. The protozoan organism is generally found in water sources, including ponds, streams, and rivers, as well as in polluted food such as raw meat. So, this is kind of a ingestive organism. It maybe affects the gut system. And Legionella pneumophilia is going to be an airborne something. So inhalation of the pollutant is the main mode of transmission here. And yeah, it's a bacterium and is observed in um, water sources, which eventually gets suspended in air as aerosol particles. And when we inhale it, we have this pneumonia or other diseases caused by this microorganism. So all of you have answered it rightly. Very easy question again. What is a fecal indicator? Can someone give what is a fecal indicator? The presence of some microorganism. Then it can be anything, right? Similar to pathogens. Are fecal indicators pathogenic? Pathogenic organisms are generally organisms that can cause disease. Okay, so we generally do this coliform or fecal indicators testing in wastewater such that the fecal indicator act as a measure to let us know whether this pathogens are present in the water or not. So we all know in domestic wastewater, or the general municipal wastewater, what happens is the content is going to be a major issue in this kind of water systems. So here, in order to identify the presence of microorganisms, what we do is we go for this indicator test, and there are certain rules or characteristics that these indicators should follow. One is that they are generally non-pathogenic organisms, and they should have the survival rate similar to a pathogenic organism. They have to be present when the pathogen is present, and absent when the pathogen is also absent. So this is very important. Maybe it may sound a bit confusing. It's just that whenever a pathogen is going to be present in water, like a fecal pathogen, a pathogenic organism that has entered the water through the feces of organisms, this indicator organism should also be present. So it should survive just in like a pathogenic organism, but it should not cause any disease, and it should be non-pathogenic itself. And also similarly, it should not be present in the water when this water is not going to contain any pathogenic organisms also. Meaning, we can check whether there is presence of this indicator, and directly this is proportional to the presence of a pathogenic organism. So it's very important that it has all these characteristics. And when we are going to detect it, we are going to know that the water is also having pathogenic contamination. In this case, salmonella, generally it's going to be Salmonella typhi, which will cause typhoid. So obviously that's a pathogenic organism. And Vibrio cholerae, it's going to cause cholera. Staphylococcus aureus is also a pathogenic organism. E. coli is the most generally used fecal indicator. And also we can also use something like a Streptococcus species, or some other coliforms. A few coliforms also are present, which can be used as indicator organisms. So yes, E. coli is the answer for this, and this is the next question. You can just give A1, B2, C3, D4, something like that in the chat box. I don't have options as in. You can just directly match and give the answers in the chat box. And also the following question will be about the defining all these terms. So in the meantime, if someone wants to try your hands on that, you can, because we'll discuss answers of both the questions together. Okay, we have a mixture of answers. I think no two answer is identical. Okay, what is a water related disease? I think we'll go with option, the D one first. We have two people telling D typhoid and two people telling D malaria. What is water related disease? What's the definition of it? Okay, so water related disease is something where we have few vectors, although these vectors act as transmitters of the disease. So this is water related disease wherein, as rightly mentioned, what okay, waterborne disease are caused. Okay, we'll discuss about that. In case of water related diseases, as rightly mentioned, we have mosquitoes. In case of malaria, we have mosquitoes where they act as vectors. They carry the um, protozoa or the causative agent for the disease. What happens is water is used for the breeding of this mosquito, not directly involved with the breeding of the microorganism. The microorganism is obviously going to stay in the gut of the plasmodium, right? Plasmodium generally causes malaria, so it's going to stay in the gut of the mosquito, and then it's going to multiply there. When the mosquito is going to bite someone, all that we get is the transmission of this microorganism into the human being, and they get the disease called malaria. So this, in water related disease, the important concept is the involvement of vectors. So whichever disease is going to have the involvement of vector, you can directly match it. So in this case, it's water related disease is going to pair up with malaria. Water is a water based disease. Yeah, vectors rely on water to complete their life cycle. So obviously mosquitoes breed in this water. So maybe dengue, malaria, all that we do is we just see there is no stagnant water near our home. Even it's been ensured by officials, they just come and visit and check if we have some stagnant water. So water is related because mosquitoes breed in them, not directly with the disease. So that's the case. What's a water based disease? Any definition? Okay, one person is saying C1. Okay, it's obviously ruled out now. C4, C3, and C4. Okay. It's between dysentery and schistosomiasis. Right. So water is going to provide an habitat for the intermediate organism, and when we are going to be in contact with that water, we are going to acquire that disease. So in this case, dysentery, I think it's out of race, or typhoid is out of race because it's not an intermediate organism that requires water as the habitat. But in case of schistosomiasis, it's caused by some worm kind of a thing, a parasitic worm, where this water acts as the habitat for the parasitic worm. So the water based disease is, I'm sorry, am I audible? I just lost connection for few seconds in the middle. Am I audible again? Okay. So the water-based disease is schistosomiasis, and in case of watershed disease, I'm sorry, it should have been water washed disease. I think that's a typing error. So waterborne disease is something where we directly ingest the pathogen through water, and in the water wash disease, it's because of lack of sanitation or lack of cleanliness that occurs because of lack of water. So in that case, the answer would be, the waterborne disease would be typhoid. Water washed disease would be dysentery, water based disease would be schistosomiasis, and water related disease would be malaria. So this is the answer. Now you can try matching this. Okay, as rightly mentioned by all of you, it's just straight. Schistosomiasis has water snails as their vector, malaria is mosquitoes, onchocerciasis is black flies is the vector for the particular disease. Okay, so this I think we have discussed with the previous question. I just added this slide so that you can have a look at all these definitions when you, in case you go through this slides sometime. The next question, I just have a couple of slides more. We'll just go through them and then. Okay, we have D and C in the answers now. Okay, I think it's pretty sure it's not hepatitis. What's the causative agent? Like, no need of the exact genus and species name. Generally, in the case of microbial classification, what causes hepatitis? What kind of microbes cause hepatitis? We'll just go through this very quickly. Virus. Okay. What causes cholera? Vibrio. What's that? Vibrio cholerae. Yeah, Vibrio cholerae in case of this general microbial terms. Bacteria, right? So obviously that's not a protozoan disease. Trichuriasis is caused by two people have answered D, right? Okay, no. Trichuriasis is caused by a worm, a parasitic worm called a whipworm. We generally call it a whipworm. So it's a parasitic worm. It's not a protozoan. But in case of diarrhea, protozoans can be involved. Diarrhea can be because of multiple um, organisms. But here we have diarrhea as the answer for protozoan disease. So here is the next question. That previous question has nothing to explain. It's just straight what's the causative organism and what group of microbes they belong to. That's it. People who answered D in that, I think you have made a note of it. It's because of whipworm. It's a whipworm infection. Okay, one has answered. So fecal indicators, um, yeah, we need not like culture them mostly, and so A and C is ruled out here, and they don't give false positives and false negatives, because like we have discussed what should be the characteristics of a fecal indicator. In the purview of that, they generally don't give false positive and false negative, so that's not a challenge for this. Insensitive, they are insensitive to disinfection than pathogen. So this is a major challenge. This is not a major challenge for a fecal indicator detection in our environmental sample. So this is the last question for today. You can just tell what sensitivity, what specificity. Think you have come across this term in week 9 bit frequently. Any answers? Sensitivity and specificity in terms of detection, in terms of um, biotechniques that we use for microbe based some techniques. Okay, so one person has answered sensitivity means detection limit and specificity means accuracy, right? Sensitivity is the detection limit, exactly. uh, in the sense how sensitive it is, in the sense, maybe let's tell I have an instrument which can detect only when the bacterial count is above 100. So the sensitivity, until 1000. So this range is defined. If I have something above 1000, maybe it can show some wrong results. If it's below 10, it can show some wrong results. So this is the sensitivity of that technique or an instrument, however we define it. Specificity means I have an instrument which can only tell me, for example, all this is just a random example that I'm giving considering the time limit. So in case of specificity, what happens is maybe I have an instrument which can only tell whether, only tell if the sample is a bacteria, or it can only act only if it's a bacteria. It cannot act with a virus, or it cannot act with some other protozoa, or the other way around. It can only act with virus but not with bacteria. So all this comes under specificity. If someone has learned about enzymes or something, specific enzymes acts on specific substrates only. For example, if I give you some cellulase, it can act on cellulose only. If I'm going to give you amylase, it can act on starch only. Cellulase cannot act on starch and amylase cannot act on cellulose. So this is something called a specificity. That's the accuracy of the target. Okay, so this is these two terms are very important, and the differentiation between these two terms is very important to know. Oh, okay, so with this, I think it's already 9 o'clock, and I think I'll discuss the question that Abhigna has asked now. I thank all others who have joined the session and who have been very interactive. I hope I was able to share few things that could have benefited you from today's session. If you want to continue staying in the meet, and if you want to ask something, you can please stay. Otherwise, I thank you all for joining in, and except Apigna, whoever wants the discussion of that question can stay, or if you want to ask something else, you can stay. Otherwise, you are free to leave the session. Thank you for joining in. I'll just go back to that question. This question, right, Abhigna? Okay. In antibiotic resistance, I guess you understood what antibiotic resistance is. Generally, we give antibiotics um, to treat microbial infections. In case of humans, we take antibiotics when we have some infection, like a throat infection or some other infection, so that this antibiotic will not allow the replication or multiplication of that microorganism, and eventually the growth of the microorganism stops and we get rid of that disease. But in some cases, what happens is this antibiotic resistance can confer the ability to the microorganism such that it can grow in the presence of that antibiotic itself. So antibiotic resistance, generally how it happens is, for example, there is some dose of this antibiotic to which the microbe is continuously exposed to. For example, I eat some 50 milligram of some antibiotic and my disease is cured. But maybe in the environment, in a water or a land sample, what happens, maybe this antibiotic can also stay and the microorganism can also stay. At that time what happens is, this microorganism get exposed to various different concentrations of this antibiotic, and eventually what happens is, at one point, it acquires this skill of breaking down this antibiotic itself. Previously what happens is, this antibiotic was blocking some or the other mechanism of this microorganism. Now this microorganism has attained a special, what to say, some ability, such that it can either tolerate the antibiotic or even break down the antibiotic, and it can happily grow in that environment. So this is the concept of antibiotic resistance. Keeping that in mind, what our first point says is, antibiotic resistance is only a problem in hospitals and healthcare facilities. uh, According to this statements, what they say is, all other places other than hospitals and healthcare facilities are not prone to this antibiotic resistance phenomenon. But that's not the case. Even in our day-to-day life, we can experience the effects of antibiotic resistance, and even all other environment other than hospitals are also responsible for conferring this antibiotic resistance. Both the ways it is true. Like for example, I have some infection and I take some antibiotic. I have some extra 10 tablets. What I do is, okay, this is not useful to me anymore, so what I do is I just flush it in my washroom, or I just throw it away in the land somewhere outside my room or my home. At that time what happens is, this antibiotics is going to be transported to a new environment, like a soil, or water somewhere else. This soil will be transported, this water will be transported, and there are going to be inherent microorganisms that are going to be present in this environment. When these are exposed to this antibiotics, all they can do is, they can acquire resistance against this antibiotic. Acquiring resistance is nothing but, they will have some special genes which help them live in the presence of that antibiotic itself. So this is called antibiotic resistance. And even when I'm going to throw out a medicine just like that, this can help the microorganisms to get the resistance, because it is continuously exposed to different dosages of this antibiotic in its environment itself. So it's not only a problem in hospitals and healthcare facilities. And one more phenomenon is, this antibiotic resistance gene can be transferred from one microorganism to another, which is even more deadly, because when this is going to be transferred across microorganisms, um, the population of microorganism which is going to have this antibiotic resistant genes is going to be very higher, which is also a potential threat. Because when, for example, when I have some throat infection or something, first I was taking some 500 mg of medicine, it was working fine for me. But after some, maybe this bacteria has gained resistance, and after 5 years or something, this 500 mg is not going to solve my problem. I should either go for 750 mg or 1000 mg, such that, okay, because of the dose of the um, antibiotic, maybe this microbe can die. But there is no guarantee, when I'm going to increase the dosage, it's going to die. Okay, so this is about the first point. About the second point is, antibiotic resistance is only natural with no human input and cannot be prevented. So this is what I told you. It's not like only natural, because we throw this medicines all right away in whichever place we can. This also has a human input to it, because we take unnecessary medicines. Uh, we give overdose for a very small infection, or we take unnecessary antibiotics for some random infection. All these are human interventions in case of antibiotic resistance uh, phenomenon. Okay, so that is also not true. Coming to the last point first, antibiotic resistance can be reversed by increasing the dose of antibiotic. Means no, it cannot be reversed. As I told you, when I take a higher dose, this microorganism can die, but that doesn't mean that the resistance that the microorganisms are acquired against this 500 mg is reversed. That's not the case. Now if I take the same 500 mg medicine, the microbe can still survive and I can still have the disease. So that point is also false. Whereas in the C point, as I told you, over and misuse of the antibiotics contribute to development of antibiotic resistance. It's just the same story giving you four different options. And in that, only C is true. Is that fine? Okay. Okay. And then I guess no more questions. Okay. Thank you all for joining in. Thank you so much.